Let's take a look at heme synthesis. Now, the process of heme synthesis is not exceptionally complex, but there are a number of steps and there's a number of enzymes that are involved. Understanding the chemistry and the enzymes is not that important at this point in your education. The most important thing to remember are the genetic diseases called porphyrias. Porphyrias are mutations in any one of the enzymes in heme synthesis. So each one of these enzymes can be subject to mutation, and this mutation will lead to a defect in heme synthesis. These porphyrias obviously can lead to profound anemias, because if you're unable to synthesize heme, or if you have a reduced capacity to synthesize heme, you will not be able to have the iron carrier for oxygenation in the body, leading to anemia. So there's a few key features that I'd like you to know about heme synthesis. First is the initial precursors. The heme molecule, the final heme molecule shown here, is a rather complex structure. But how we get to that structure is rather simple. We start out with two precursors. We start out with succinyl-CoA, which is obtained in the TCA cycle. And we start out with the amino acid glycine. Now, in order for a TCA intermediate succinyl-CoA to be a substrate in heme synthesis, it's localized in the mitochondria. So that's why heme synthesis involves both mitochondrial and cytoplasmic enzymes. And there's a passage between these structures. So we take these two small molecules, succinyl-CoA and glycine, and we use the first enzyme of the heme synthesis pathway, and that is ALA synthase. ALA stands for aminolevulinic acid. Aminolevulinic acid is the first product of this pathway. Now, if you notice what happens here, we take two of these aminolevulinic acid structures, and we add them together with the dehydratase enzyme, and we close up a ring structure. What we get is the first ring structure of the heme molecule. Ultimately, the heme will have 1, 2, 3, 4 parole structures. Here's our first parole. The next step of the reaction is to take 4 of these, link them together in a linear fashion. Then the third step is to close the ring so that now we have our porphyrin ring structure, which is what we begin to recognize as fully synthesized heme. And then the next several reactions are simply modification reactions that perform chemistry on some of the side chains of the heme molecule. These side chains are important for the heme structure to bind into the active sites of proteins, such as the globulins, myoglobin, and hemoglobin. The final step, once we have our protoporphyrin structure, proto means incomplete, we're going to complete it by adding iron. And the iron is going to be in the reduced 2+ state. So this is ferrous iron, which is iron 2+. This is going to be added to the heme structure using the enzyme ferroketolase. So this is our last step. to synthesize the final heme structure, which can now be introduced into protein active sites. The two other pieces of information is lead. Lead, abbreviated Pb, has an inhibitory function on two enzymes in heme synthesis. The first one is aminolevulinic acid dehydratase. The second enzyme that lead inhibits is ferroketolase. Now, the significance of this is lead poisoning. Lead poisoning will lead to inhibition of these enzymes, which essentially inhibits heme synthesis, leading to anemias. So this is the mechanism of how lead poisoning leads to anemias.